Okay, I think we can go ahead and get started. All audience questions will be answered at the end. And so if you have questions throughout the presentation, you can type into the Q&A box, or at the end of the presentation, you can raise your hand and your mic can be opened so you can ask questions live. And with that information, I'm going to introduce MJ. MJay Espina is a second-year PhD student with Drs. Aaron Lorenz and Robert Stupar. MJ obtained her Bachelor of Science degree in agriculture, majored in plant pathology from the Visayas State University in the Philippines. After finishing her undergrad, she worked at the Philippine Rice Research Institute as a science research specialist on the Golden Rice Project. This project made an impact on her and led her to pursue a graduate degree in plant genetics. She earned her MS in agricultural science, major in plant science from Tennessee State University, with her research on developing soybean mutant populations. In 2017, she joined Dr. Aaron Lorenz's lab as a research technician, working on introgressing high oleic and low linolenic traits into Minnesota elite soybean lines, and later started her PhD in fall of 2019. Today, she'll talk about her PhD research on iron deficiency chlorosis in soybean. Outside of graduate school, MJ enjoys spending her weekends baking, biking around state trails, exploring outdoors, hiking, and playing tennis with her husband and son. So take it away, MJ.
Thank you so much, Kate. So today I'm going to talk about identification and characterization of candidate gene conferring resistance to iron deficiency chlorosis in soybean. So I'm working with Drs. Aaron Lorenz and Dr. Robert Stupar on this project. So the flow of my presentations will begin by talking about soybean and iron deficiency chlorosis, followed by my research objectives. And then I will talk about the experiments that I'll be doing to address these objectives. And finally, just talk about the summary of my presentation. Soybean, or glycine max, is a legume species originated in Southeast Asia. So this is how soybean looks like in the field if you haven't seen soybean before. It is a self-pollinated species. It's a diploid with 20 chromosomes. And it was domesticated from its wild ancestor, glycine soja, in China around 3,100 years ago. And this is how the domesticated soybean look like today, mostly yellow seed coat. And this is how the wild soybean look like. So for a crop that has been domesticated around 3,000 years ago, we made a lot of improvement in soybean. Before going deep into my talk, I want to talk about how soybean was introduced in the US. It was first introduced in the US in 1765 in Savannah, Georgia, originally in a small farm for soy sauce production, but that business died out and there's no long-term impact in terms of growing soybean in the US during that first introduction. And a little bit less than a century later, around 1851, soybean was reintroduced in the Midwest, specifically in Illinois, and then it was planted on states around Corn Belt. And in 1870s, it actually increased its popularity among farmers due to use as an emergency forage crop or an animal feed. And then in 1904, George Washington Carver, he is a very famous chemist, and he is actually very famous for his work on peanut, but he also worked on soybean and discovered more valuable uses of soybean, such as protein and oil. And in around early 1900s, there were only 20 soybean varieties that are widely planted by the farmers. And the first president of the American Soybean Association, William Morse, went to China during that time and collected around 10,000 soybean germplasm, which was the core collection of the soybean germplasm that we have now. So if you can see the progress of the soybean production in the US, it's not actually like widely planted right away. So there's a slow progress of production. And in post-World War, in mid-1940s, they discovered more uses of soybean, such as protein and oil. And that's when the universities or establishment of the breeding programs in the US universities started. And just to give you a fun fact, the first soybean breeding program in the University of Minnesota was established in 1945 with Dr. Jim Lambert as actually at that time, part-time soybean breeder. And since that 1940s, it skyrocketed, and soybean now that we know is like second most widely planted crop in the US. So with that, I'm going to transition to the uses of soybean. So soybean is actually composed of 80% meal and 20% oil. So 97% of that meal is used for animal feed, and small portion of that, which is 3%, is used for soy food or other soybean protein alternative products such as tofu. And on the other side of that, 20% of oil were used as food oil, and then some of that were used as biodiesel oil, and a small portion of that were used for industrial purposes such as crayon, paints, and cleaners. So in 2020, soybean is the second largest crop in the US, with a total production estimated to 4.14 billion bushels. For Minnesota, in 2020, there are about 7.4 million acres planted in Minnesota, with the acreage concentrated in the northwestern part of Minnesota and the area surrounding the Red River Valley. The 2020 production of soybean in Minnesota is around 359 million bushels. With these statistics, Minnesota ranks as the third largest soybean producing state in the US. However, there are factors that limits the yield of soybean into reaching its full genetic potential. So some of these factors include soybean cyst nematode, iron deficiency chlorosis, aphids, Phytophthora root rot, white mold, sudden death syndrome, some seedling diseases such as Rhizoctonia, Pythium, and Fusarium. These are fungal seedling diseases. If you notice, among all these stresses that impact soybean production, iron deficiency chlorosis is the only abiotic stress that is mentioned here, and it has a major impact on soybean production. So with that, I will transition my talk into iron deficiency chlorosis. But before going to iron deficiency chlorosis, I want to talk about iron first. Iron is an essential micronutrient. It is the most abundant micronutrient in the soil. It is available in two forms. The iron 2+ or the ferrous form of iron, and the iron 3+, which is the ferric form of iron. So these two forms are very important because they are reversible forms, and one form has better solubility over the other. So iron 2+ is more soluble than iron 3+. And then iron is also important in basic plant function, such as chlorophyll synthesis, respiration, and nitrogen fixation. So here, this is an idealized image of chlorophyll synthesis, where you start from nitrogen and then the plant synthesizes the chlorophyll and undergoes different steps. And within that different steps, iron right here is an important part of chlorophyll synthesis. And then another important function of iron is for energy production. So right here, this is the mitochondria, where you can find iron specifically in cytochrome, and it is very important in energy production of the plants. So as I mentioned in earlier slide, iron is important requirement of chlorophyll synthesis. So with this lack of iron or iron deficiency, you can see symptoms such as intervenal chlorosis. So you can see here that there's yellowing of the leaves and stunting. So this is due to, as I mentioned, chlorophyll synthesis. Iron is important in chlorophyll synthesis. That less iron, less chlorophyll synthesis, that does less photosynthesis. So we can observe yellowing of the leaves due to lack of green pigment chlorophyll. But where do we find IDC or iron deficiency chlorosis in the environment? So IDC is prevalent in alkaline or high pH soils greater than 7.5 and calcareous soil. So this is soil pH map of Minnesota. And if we go back to the counties that are planting soybean in Minnesota, if we can see here the northwest part of Minnesota. So if you can compare that to the pH map where the reds are acidic and the greens are basic. So in this region, it is mostly on that green color. But you might think, where did the calcium carbonate came from? So a little bit of history here. Millions of years ago, during the Cretaceous period, actually, this is Minnesota or US, mostly US, is a part of an ancient seabed called Western Interior Seaway. So it starts from Gulf of Mexico going up north to now US and Canada. And if you can see here, Minnesota is sitting right here, which is this region where soybean are mostly planted. And the reason why this is high in calcium, because if it's an ancient seabed, it has some seashell deposits, which is very high in calcium. And this problem is not prevalent probably when you talk about 40 years ago, because this area in the northwestern part of Minnesota were planted with oats and barley and wheat. But now soybean production actually starts to move north in the last 30 years and going west, such as Nebraska and North Dakota. And this iron deficiency is also a problem in those regions. So with that, I want to talk about the economic impact of IDC. So the yield loss estimate based on our latest information, which is not very recent, this is based on 2012, is around 260 million yearly losses due to IDC. Just to give you an idea of how much soybean contribute to US economy, soybean is the total production of soybean in 2020 is estimated to be $46 billion. When you think about it, soybean is widely planted in all over US, but this is a regional problem. So that $260 million losses, when you just look at the upper Midwest, so going back to individual farm, it could be a big percentage of loss. So how can we manage IDC in the field? The first line of defense of IDC is using resistant soybean variety. Just to show you how this resistant variety works. So on the left right here, this is a susceptible variety, and on the right here, this is the resistant variety. And if you can see, there's the difference with the appearance. So you can see the susceptible variety is very yellow. Other management strategies include use of companion crops such as oats, reducing plant stresses, so reducing that diseases or stresses due to herbicide spray, increasing plant density, and use of iron chelate. Now that we know that using resistant variety is very important in management of IDC, so how does plant cope with IDC? So there are actually two strategies for iron uptake in plant. So the first strategy, which is common to dicots and ungraminaceous monocots. So here is an idealized image of rhizosphere, cell membrane, and the cytosol in the roots. So for strategy 1, the mechanism is actually the plants actually excrete these protons, and these protons will acidify the rhizosphere, and once it became acidic, this iron 3 plus, or the ferric form of iron, will be reduced to iron 2 plus, which is available to the which is available to the plant. The second strategy for iron uptake in plants is strategy 2, which are common to Graminae and other cereal species. So again, this is the idealized image of the rhizosphere, the membrane, and the cytosol. So how this works is the plants release the phytosiderophore. This phytosiderophore will form a complex with iron 3 plus, and this complex, the iron 3 plus and phytosiderophore complex, will be transported inside the plants. And once inside the plants, the iron 3 plus will be released and will be mobilized in the plants. So now that we know how plants deal with iron deficient condition, so just a reminder that soybean exhibits strategy 1 resistance mechanism. So we know that roots are very important. It is important in the proton extrusion to acidify the rhizosphere right here. But there are still questions on other plants of the plants, such as stem. So are there any other molecules involved in iron transport? Can seeds serve as iron storage? So these questions are the type of questions that I want to look at in my research. But before going further, I want to talk about the mapping studies that has been done related to IDC in the past years. So this is just a summary of different QTL mapping studies done in IDC. Just to have a little background here. So these are soybean chromosomes 1 to 20, and these are the position in megabase per. So here in earlier mapping study, if you can see that the interval are quite large. So this is not due to the interval of the QTL that are very large. This is due to lower resolution mapping, because in early 2000s, so SSR markers are very common. So, but when you can see that smaller interval, so this mapping population use a high density marker mapping. So I just want to focus here in chromosome 3. So chromosome 3 is very famous IDC QTL that's found in almost any mapping studies. But for this research, I want to talk about mapping study done by Mary et al. 2020, where they found a IDC QTL in chromosome 5. So I want to talk about how this population was developed. So the chromosome 5 QTL was developed using one of these parent, FISCB3, which is VI438471. It was originally a Swedish edamame variety. Of course, it's originated from Sweden. It was bred by Dr. Sven Holmberg for actually protein. This is very high in protein. So just a small anecdote on this variety. This variety was brought here to America during war because this edamame variety is very high in protein. So this serves as their protein source, and it thrives in northern climate. It's very early maturing varieties, which just matures around 75 to 80 days. So we use this as a donor for mapping IDC resistance QTL. So in addition to IDC, it also exhibits tolerance to drought, ozone, salinity, and aluminum. Another parent that we use for this biparental mapping study is the Mandarin Ottawa, or the PI 548375. So a little bit of background about North American soybean germplasm. So 85% of North American soybean germplasm actually originated from a very narrow genetic base. So it's just comprised of 18 ancestors, with Mandarin as top 2 contributor of this 85% of North American germplasm. So we use Mandarin because of that reason. And also Mandarin is highly adapted to northern climate. And then these two variety were crossed. So Fiske B by Mandarin, Fiske B as a resistant toner. This was done by Carl Butenhoff. He was a student with Dr. Jim Orff. Dr. Jim Orff was the previous breeder here in University of Minnesota. So he developed recombinant inbred lines and then course mapped the IDC QTL. And then from the recombinant inbred lines, he found some materials that are still segregating at chromosome 5 QTL, and then developed near isogenic lines for that. So just a background for non-breeders, near isogenic lines are you have basically the same background, theoretically the same background, except for the region of interest that you only, the difference is the region of interest. For example, on one part you have the susceptible type, and on the other part you have the resistant type, and the rest would be susceptible. So as a result of that biparental mapping study, Mary et al. 2019 reported that there's QTL found in chromosome 5. So right here on the x-axis are the chromosomes, and the y-axis are the load scores. So they found a QTL in chromosome 5, and they actually found another peak in chromosome 6. And then Dr. Ryan Mary, who's a previous graduate student to work on this material, he actually validated that QTL that Butenhoff 2015 found, and he used two association panels. So the first panel he used is the 1,336 diverse accession. These are composed of different land races and some germplasm from soybean collection. And what he found is he found that classic chromosome 3 IDC QTL, but also he confirmed the QTL found by Butenhoff 2015, this chromosome 5 QTL. And then he actually looked at another panel, which are composed of 854 advanced breeding lines. So these advanced breeding lines were the lines as part of the Northern Soybean Regional Trials. So the breeders from northern states, so they have their advanced breeding line sent to regional test, and these materials were tested for iron deficiency chlorosis. And what's very interesting about this QTL is this locus is present in elite lines, but the chromosome 3 locus, which is very popular in IDC, you cannot find it in the elite or advanced breeding line. So there's the value on working on this chromosome 5 QTL because it's still segregating among elite breeding lines. And then Mary et al. Further did fine mapping of the biparental QTL that was found by Butenhoff 2015. And first, they course map it to 1.4 megabase, and then one round of fine mapping, Mary et al. 2019 narrowed it down to 450 kb, and another round of fine mapping, he narrowed it down to 160 kb, and lastly, he narrowed it down to 137 kb region spanning 17 genes, and out of those 17 genes, we have a putative candidate gene in this region. So these materials, I will continue. This is the material that I've been working on, and this candidate gene is also the candidate gene I've been working on. So with that, I will talk about this candidate gene, MATE efflux, our candidate gene within that region. So MATE is a multi-drug and toxic compound extrusion. MATE is a member of transporter family proteins. Some known functions include hormone signaling, aluminum tolerance, and iron homeostasis. So just an example for function as for iron homeostasis, this right here is comparison between Medicago trancatula wild type and a mutant, where it's like a defective mate gene. And what they found out is when they put it under iron deficient media, they can see this chlorotic symptom, which they cannot find in the wild type counterpart of that. Also, when we look at that 17 genes, one of our candidate gene is the mate efflux. This mate efflux is actually very similar to mate transporter also found in Arabidopsis, which mediates the iron homeostasis. So what they have right here is a mutant of BCD, which is a Bush and chlorotic dwarf gene. So they put it under iron deficient media. And if you compare the wild type and the non-mutant BCD, they are green. And when you look at the mutant BCD, it's actually chlorotic. And when they feed iron to that the phenotype was actually recovered. So it now turns green. So this makes us hypothesize that this mate candidate gene is really strong candidate gene within the region. So with that, I will have a recap for what I just talked about. So soybean is very important crop in Minnesota. So annual yield losses estimated to be $240 million. And using resistant variety is very important to manage this abiotic stress. And previous students from these labs mapped QTL in chromosome 5 conferring resistance to IDC. And they narrowed it down to 137 kV region spanning 17 genes. And within that region, we have a very strong candidate gene. And then I will transition to my research objectives. So for this research, the first objective is to identify the causal gene by candidate gene association mapping and fine mapping. And then we also want to validate the candidate gene by expression analysis and targeted mutagenesis. And lastly, we want to determine the effect of this QTL on seed iron assimilation across different plant nodes. So for my experiments, before going deep to my experiments, I want to look to talk about the resources that are available to me. So first, I want to talk about the plant materials. The soybean research community actually have this resource, which we have 481 resequence accessions. This composed of very wide accessions spanning from elite lines, land races, and Piscivetry is one of those 481 accessions. And then, of course, as I mentioned earlier, I will also use the 137 kV near isogenic lines developed by Mary 2019. Another resource on this project is Mary 2020 developed this growth chamber assay. So as I mentioned in my introduction, that the field condition that actually triggers this iron deficiency chlorosis is high pH and high calcium. Mary 2020 developed this assay where he adjusted different pH levels and different calcium carbonate concentrations, and he developed this assay. The reason behind developing this assay is the IDC nursery, which we do our phenotyping, is 3 hours away. And doing some molecular studies and physiological studies, sometimes you have to deal with RNA samples, and it takes 3 hours to go to the field and then going back to process those samples. So having this controlled environment assay is very useful in further exploring this trait. And we use IDC rating scale, both for field and growth chamber assay, with 1 as the resistant. and 9 as the chlorotic or necrotic or almost dead. So it's like a range of phenotype. So although that we have this growth chamber assay, there is still a value of looking the actual field because, looking at the actual soil condition or the actual field condition is really important in this case, because although we know that iron, pH, and calcium carbonate is the major driver of this abiotic stress, there might be some other factors that we can find in field environment that we cannot mimic in the growth chamber assay. So all my materials will also be phenotyped in field, and we will use visual rating following this 1 to 9 rating scale. And former student from our lab, Doubles 2019, developed this pipeline using UAS, or Unmanned Aerial System, or in layman's term, drone. So we use drone in our breeding program to phenotype iron deficiency chlorosis in the field. So I will also use this phenotyping, so looking at both visual and the drone rating to phenotype my IDC materials in the field. The advantage of using this drone is it's very time efficient, as well as you remove that subjectivity in visual rating. So you will only have the objective rating, which is what you actually see in the field, like in the eyes of the camera. Other tools that are available to me, I will also use some gene expression analysis. So for this research, I will just use QRT-PCR. And then we will also utilize hairy root transformation, because when we look at our trait of interest or our gene, we look at the gene expression atlas, and these genes is actually expressed in the roots. So we think that hairy root transformation is a good way to start when we are just looking preliminary experiments of this gene. So we will do overexpression and gene localization using hairy root. The advantage of hairy root is it's very quick and easy. So you can have your phenotype 1 month after you start doing the transformation. So I will also utilize CRISPR-Cas9. So it is a gene editing tool that we do to have mutagenesis on a specific parts of the genes. So it's a targeted mutagenesis tool. And lastly, I will use an Ionomic Analysis tool. So Ionomic Analysis, if for you those who are not familiar with Ionomic Analysis, so if you look at genomics, so when we talk about genomics, we talk about the entire genetic content of the plant or any other species. So when we talk about ionomics, so instead of solely looking at the iron content or iron concentration in a seed, for example, with ionomic, you can have the global or the entire ionome profile, which consists of, in this case, we will look at 20 different elements. So the reason behind using ionomic instead of analyzing a single element or just looking at iron, because in actual field condition, other elements such as phosphorus or aluminum plays an important role in that interaction with iron. So those elemental profile dynamics, we want to capture that by using ionomic analysis. So with that, I will talk about the objectives and the experiments that I will be doing to address these objectives. So the first experiment I will be doing will be to satisfy first objective, which is identification of causal gene within 137 kb region. Although we already have putative or hypothesized candidate gene, we still want to narrow down that region. So just to have more evidence pointing to that candidate gene. So we want to do a candidate gene association mapping. So here we will utilize the 400 plus diversity accessions that I talked about earlier. And since this accession has available sequencing data at depth of 15x to 40x. So we want to analyze the haplotype within our region of interest, basically the 137 kV region, and understand the diversity. We want to look at the rate of LD decay, and determining the rate of LD decay will give us, it will provide us if we have enough mapping resolution. And if the LD decay within the region is very low and variants are not correlated, then we can decouple these different variants and hopefully map our candidate gene. And then we will use mixed linear model for this association mapping. And as with the traditional association mapping, we will account for population structure and family relatedness. The second experiment for this identification of causal gene, going back, so those materials will be phenotyped in IDC nursery. It will be just for this season, it will be just in one location since we have very limited number of seeds with 2 replication following randomized complete black design. So the second experiment is having this one more round of fine mapping, since within that 137 kV region, we still found families that are segregating. And we want to genotype this and look for more recombinants to narrow the region. So this is just an initial results, what I got. So last summer, I planted these different heterogeneous families in the field, and within that 137 kV region, so these are the marker locations. So I have this scattered, I have 4 markers scattered across that region. So 27, 62K, 102K, and 130K. And these are just examples of the different recombinants that we found. So we found that there's a FISCB in this half of the side, and some of it FISCB on the other side. So what we want to do with these recombinants is actually phenotype it in the IDC nursery, again using one location to replication and following RCBD. So this is what an ideal recombinants would look like. So if this is our region of interest, we have this recombinant A, which is mandarin all the way. So the IDC phenotype in the field you would expect would be susceptible. And if this is FISCB all the way, except for the region of interest, which is mandarin, we also expect to have susceptible phenotype. And for recombinant C, which is mandarin all the way, but FISCB on the region of interest, so we will expect a resistant IDC phenotype. And this is just a confirmation that if it's FISCB all the way, we will have a resistant phenotype in the field. So with these different recombinants that we have, we will hopefully tile this region based and associate the phenotype with the genotype, and hopefully narrow down the region into a handful of genes. So as a recap, we have 2 methods to identify our causal gene. So we have the candidate gene association approach and the fine mapping approach. All the materials will be phenotyped in IDC nursery in Danvers, Minnesota. And due to the limited number of seeds, experiments will be conducted in one location during first year. The second objective of this research is the validation of candidate gene. So the first experiment for this validation objective is the gene expression analysis. So when Mary et al. Sequenced the parents, FISCB and mandarin, we found out that our region of interest actually has a data box insertion in the promoter region. So data box is a signature sequence found in a promoter. It is where the transcription machinery will bind, and we hypothesized that an increased, or if there's an insertion, you have extra data sequence in that region, there could be an increase in expression. So Mary 2020 did a preliminary study looking at the gene, the mate gene in that region. So on the y-axis right here is the fold expression difference of the gene, and They look at week 1 and week 2. And then on the x-axis right here are the different Fischb and Mandarin combinations. So this this expression right here was based compared to Fischb3. So for example, Fischb3 iron deficient condition compared to the Fischb3 with iron. So there's an increased expression in the Fischb3 iron deficient, and we didn't find that same increase in expression in the second week. So this is just to compare Fischb Mandarin an iron deficient condition with the Fischb in the iron sufficient condition, and you also didn't find that increase in expression. So with this initial result, we want to further validate this experiment by planting these materials in the growth chamber, where we plant the Fischb tree and the mandarin. And then after 7 days, we will apply the nutrient solution at pH 7.5, 7 days after planting. I'll treat the seeds, the plant with 0 CCE, basically no stress, and 30% CCE, or this is calcium carbonate equivalency. So we are raising the pH at the same time we are raising the calcium carbonate. So this will be replicated 4 times with the pots as an experimental unit following completely randomized design. So we will also look at this at different time points. So 1, 2, 3, 6, 9, 12, and 15 days after treatments, because we found out that the first week, there's an increase in expression, but we did not find the same increase in expression in the second week. So with that, we want to know when this is exactly expressed, so we know the plant response, the exact time of the plant's response to IDC. The second part of this experiment is overexpression of mate gene in hairy root. So here we attach the 2 dose of 35S promoter to our mate gene, and we want to look at the iron accumulation in the roots and the actual expression. So the hypothesis behind this experiment is overexpressed mate can uptake more iron, which could lead to either iron toxicity or accumulation of iron in the roots. So with that, we have a very preliminary result. So we did a hairy root transformation of the Williams 82 background, which doesn't have the Tata box insertion. So when we put 2 doses of 35S, so we expected that there's an overexpression. So this is transform roots and the iron plus and iron minus media. So this part of the study, the microscopy work was done by Bruna in our lab. So what we found here is the transform roots under iron condition, we see this black staining. So we use iron staining in this experiment, which this black staining indicates some iron localization. So what we found out that is there's an accumulation of iron in the apoplast. So what literature says about this is that apoplastic iron storage is a form of an short-term storage, and it will decline over time as the iron is mobilized to the upper parts of the plants. So this result is very preliminary, and we don't have enough replicates for this to really finally conclude. So with that, we want to design more experiment. So we know it's in apoplast, but really which part of the cell, we don't really know where that is. So we want to do we want to know where the gene is expressed and localized. So in order to do that, we want to attach the native mate promoter to our reporter gene, for example, MJay. So I'm just putting an example of possible reporter gene that we will use. And we will also do gain of function. So instead of just looking at the roots, we want to overexpress this mate gene in a whole plant in the background of Williams 82, which doesn't have the Tata box insertion. So overexpressing these genes, we might observe some leaf phenotype that could explain some of the mechanism of this trait. And then lastly, we want to do targeted mutagenesis using CRISPR-Cas9. So for this research, we want to knock out MATE gene. So the background that we want to use for this is the FISCB, since FISCB3 already have this data box insertion, which is our putative cause, hypothesized cause of this increased expression. So we will do it using whole plant, of course, and this is just a picture of tissue culture that could be done. So this is just a plan for now. This part of the experiment hasn't been started yet. The hypothesis of doing this is if the MATE gene will be knocked out, there will be loss of function. And with that loss of function, we want to see like changes in response to IDC. So with this targeted mutagenesis, this could give us a really strong evidence. If we have loss of function and there's a change in response to IDC, this would give us a strong evidence to point out and to validate this candidate gene. So to recap, we will use 3 approaches to validate our candidate gene. So first, we will use gene expression analysis, and then we will also use gain of function or overexpression. We will also use loss of function validation or the knockout. Initial result shows that there's an iron accumulation in the roots, but the localization where the gene is expressed or specifically expressed within the cells are still unknown. And lastly, my third objective is determination of seed iron content by Ionomic analysis. So here we want to look at the near isogenic lines, the susceptible and resistant lines. So we will plant it in the field, and then we will get the single seeds, and those seeds will be weighed. So to correct for the different seed size, seeds will be digested and measured using ICP-MS, and we will have the data for 20 different elements. So the main purpose of this is to understand the ionomic traits in the actual field condition with or without IDC stress. So if this will affect how much seed is being accumulated in the seed. And then this part of the research, the ionomic analysis, we don't have the capacity to do this in our labs. So seed samples will be sent for ionomic analysis to Baxter Lab in Danforth Center. And then the field design for this, so we have 2 locations. So we have the IDC stress conditions, or basically in our IDC nursery, and another location is the non-IDC location, just as the normal yield field. And then these are planted in 3 replications over the 2 years, following randomized complete block design. Over et al. 2016 actually found out that there is a difference in seed compositional traits in soybean at different canopy positions. So at the bottom, the seed compositional traits are higher in amount compared to the top part of the canopy. So in order to address this variation, we want to do subsampling. So for our subsampling, we will sample 3 plants per plot, and within each plant, we will sample 3 nodes per plant. So the representative like bottom node, mid, and the top nodes. So to wrap this up, what we just talked about, so we have this 137 kb fine map region spanning 17 genes. And we want to narrow this region down to a handful of genes. And we have a very strong candidate gene within the region that could be the causal gene for the increased IDC resistance. We want to characterize these genes by looking at the expression, localization, and functional validation. And lastly, we want to look at the effect of this QTL on the changes of seed iron accumulation under normal and stress condition. So with that, I want to thank my advisors, Dr. Aaron Lorenz and Dr. Robert Stupar. Also my committee members, Dr. Katie Hirsch, Dr. Walid Sadok, and Dr. Sean Cortin. I also want to shout out to members of Stupar Lab, especially to Dr. Ryan Mary and Bruna. Also, I would like to shout out to members of Lawrence Lab, so especially to my undergrad students that worked with me over these last 2 years, Tanis Anderson and Alina. And of course, this project would not be possible without the support of our funding source, Minnesota Soybean Research and Promotion Council and United Soybean Board. With that, I will take any questions.
Great. Wonderful job, MJ. That was fantastic. So now is the time where if anyone wants to submit questions, you should. We have one so far from Don that says, were some of the soybean rows in the introduction dicamba injured?
So are you talking about the first one?
Yeah, I think this was from the very beginning of the presentation.
Oh, yeah. So this was actually the picture that I took is not an IDC, it's just a regular soybean, since at that time I was just talking about soybeans. So this is actually in our crossing black last year. And yes, we have that herbicide injury.
Hi, this is Don. Just one one question. How good are the soybean lines that are resistant to iron chlorosis?
So
Are the majority of the lines that have been produced resistant, or do they carry a yield drag?
So yeah, so if we're so I think I can only answer in terms of this. I don't know really about the other IDC QTL locus, so I will just talk about the chromosome 5 that we're working on. So this chromosome 5 QTL is has been backcrossed to our elite lines with susceptibility to IDC, and we actually observe a 1 point. This is as early as backcross through 2. We observe an early one like increase in score. So 1 point difference between susceptible and resistant. And in terms of yield drag, we don't know for sure. There could be, but we don't have enough experimental data to support that. But these backcross lines are now being planted in the field, and we will gather yield data. So we don't have enough evidence to support if there is a yield drag. That could be a possibility.
But out in the commercial world, how many of the varieties have resistance to IDC?
So there are a lot. So in our breeding program, there are actually some materials that are being sent to us by like private industry as our part of public and private breeding program. And we have materials that are actually have rating scores of 1. And even in our own breeding program, we have this line that is very resistant to IDC, but it doesn't even carry the QTL that I'm working on right now. So it's like 1. So 1 is the very resistant. So it's usually around 1 or 2, but it doesn't have the QTL that I'm working on. So that would be interesting to look at this like pyramid, just hopefully. But yeah, and also like IDC scoring also varies from year to year. So we have that variation. Some of the year we really have a good phenotyping because there's a good IDC pressure in the field, but sometimes they don't. And if you happen to test those lines in that they say resistant in a good year, you can actually do that, but you can actually see the actual performance. But if you happen to see the line that they're claiming resistant in a bad year, so we can definitely tell how resistant they are.
I have one more if no one else is going to come on.
Fire.
So, so you listed cover crops as a mechanism to reduce IDC. What is that mechanism? How does that work? How do cover crops
Yeah, so basically we term as companion crops or cover crops. So basically it's just taking up that excess nitrate in the soil. So but for example, the oats, they don't actually grow it until maturity. They just grow it just enough and then they carry it out, because if they just to reduce that soil moisture and bicarbonate buildup, because if you grow oats, for example, as a companion crop and you grow it until maturity, there's a possibility that when you plant your soybean, you don't have that enough moisture in the field. Thus, you will have another problem, which is like drought.
Okay, we do have one more question. And they ask, what are the implications of your research for plant breeding?
So yeah, first, there has not been lot known about IDC in soybean, because as I mentioned, it wasn't until like 40 years ago that the planting actually shifted to the north. And there are so many IDC QTL study, but no genes has been cloned. So cloning this gene, which gene is actually responsible for this increased resistance, actually will give us insight of the mechanism, both molecular and physiological mechanism. And this could not only be applied to soybean, but it could only be applied to other crop species that has iron deficient chlorosis with the same mechanism. And another thing that I want to add to that is we are using this QTL as our donor to introgress the trait to our breeding lines, and we started doing that. So that could be one impact of this QTL, is to eventually bring it to farmers' field.
Okay, someone else, or we have a couple more questions now. Someone else would like to know, which software are you using for your candidate gene mapping?
So the candidate gene mapping. So I'm gonna be using Tassel to analyze the LDDK, but I haven't actually done it yet, the entire analysis thing. So I'm not sure what are the other software that I will be using, but for sure, initially to get that rate of LDDK, which is the first thing that I will do, I'm gonna use Tassel.
Okay, and then Rex would like to know, if the gene is found useful, are there plans for you to get a patent on the gene?
So I don't actually have an idea. That could be for sure a discussion within our group, probably with Aaron and Bob.
Okay, we can probably wait a couple more minutes to see if any more questions come in. But we are coming up against our time limit at the moment. So if anyone has any more questions, now's the time. All right, Vinny would like to know, how are you selecting sites for gene editing?
Oh, so there are actually two types for our gene editing. So since we know that FISCB allele has that insertion in the promoter region, so the first plan is to target that region and having that promoter allelic series. So if we can see like variation in response. And the second part is like just knocking out the first exon, because usually in our group, knocking out the first exon actually works really well in knocking out the gene.
Okay, if those are all of the questions, I think we can probably start wrapping up. Thank you so much, MJ.
Thank you all for tuning into my seminar and for everyone who attended. I have some friends all the way from Norway who attended this seminar, so thank you so much.